Role of nucleotides. Nucleic acids, metabolism, enzyme cofactors, biosignaling. Role of DNA. Storage of biological information. Role of our RNA. Ribosomal component, protein synthesis. Role of mRNA. Copy of genetic information. Role of tRNA. Delivers amino acids during protein synthesis. Structure of nucleotide. Nitrogen-containing base, purine or pyrimidine, pentose, ribose or deoxyribose. Phosphate group. Nucleoside. Base pentose. Difference between pyrimidine and purine. Purine has 2 rings, pyrimidine has one 5-membered ring. Bases in DNA versus RNA. DNA, A, G, C, T, RNA, A, G, C, U. Adenine. Purine found in DNA and RNA. Guanine. Purine found in DNA and RNA. Cytosine. Pyrimidine found in DNA and RNA. Thyma. Pyrimidine found in DNA. Uracil. Pyrimidine found in RNA. The blank determines the type of nucleotide. Pentose. How are ribose and deoxyribose different? Ribose, 2 O groups coming off of sugar, deoxyribose, 1 O group coming off of the sugar. Nucleotide variants. Phosphorylation variants, role in RNA hydrolysis or biosignaling, variants of purine and pyrimidine bases, role in gene regulation. How do DNA and RNA link with each other? In DNA and RNA, nucleotides are linked by a phosphodiester bond connecting the 3-carbon of the upstream nucleotide with the 5-carbon of the downstream nucleotide sequence in 5'3". In vitro, in alkaline conditions, RNA hydrolyzes rapidly but DNA does not. Why? The hydroxyl group on 2-carbon, especially in an alkaline environment of RNA, can sever bond and form cyclic structure. RNA hydrolysis. Non-enzymatic, especially under alkaline conditions. Properties of bases. When free, are weakly basic, aromatic ring, electron distribution and UV light absorbance. Hydrophobic at neutral pH, base stacking, important in 3D structure, base pairs. Image of how base pairs bond. No, they are different species. What if you isolate DNA from different tissues of the same rabbit or from different rabbits of the same breed? Would you expect different or equivalent adenosine composition? It would be equivalent because DNA is in every cell. What if you isolate two DNA samples from the same rabbit but 1 year apart? Would you expect different or equivalent adenosine composition? Same, because age doesn't change DNA makeup for the most part. When you compared the nucleotide composition of the rabbit and of the mouse DNAs, you found that they are different. However, the amount of purines was equal to the amount of pyrimidines, not only within the individual species but also between the two species. Why? Because purine must bind with a pyrimidine. Chargaff's rules. Percentages of adenine equals thymine and cytosine equals guanine in a DNA molecule. Nucleic acid structure. Primary, nucleotide sequence. Secondary, the stable structure taken by the nucleic acid. Tertiary, folding into chromosomes or chromatin. The double helix. Right-handed double helix, 10.5 NuCl, turned backbone, out, bases went in, perpendicular to the axis, minor and major grooves, stabilized by hydrophobic stacking and base pairing. Antiparallel. The rotation of how many bonds determines nucleotide conformation. 7 bonds. Nucleotide conformations. 2 conformations permitted with respect to the base, syn and anti-purines, both. Pyrimidines, anti. What type of charge is DNA attracted to? DNA is negative, so is attractive to a positive charge. DNA forms. A form, in some solutions, usually only observed in test tubes, grows not as pronounced. B form, most stable, by far the most common in the cell. C form, left-handed. Purines are in syn conformation altering, with pyrimidines in anti-conformation, not often believed to have a role in gene regulation. Types of unusual nucleic acid structures. Palindrome, mirror repeat, hairpin, single strand, cruciform, double strand. Triplex DNAs, tetraplex DNAs, G tetraplex, high proportion of G residues in palindrome. A sequence which is followed by its complement. Mirror repeat. Hairpin. Single strand. Cruciform. Double-stranded. Of hairpins and cruciforms, which are double, single-stranded. Hairpin, single strand, cruciform, double strand. RNA 3D structure. The single-stranded RNA assumes a right-handed helical structure. It can pair with RNA or DNA in anti-parallel manner. The 3D structures of the RNAs are unique, form double-stranded structures with itself, hairpin, internal loop bulge, usually in A form, C form only in the lab. B form has not been observed. Stability of RNA. Base pair stacking, G equals U pairing, only in R and A, during folding, H bonds. Blank determines RNA function. Specific structures. Nucleic acids are inherently blank. Stable. Denaturation, aka melting. Extreme pH and heat can change DNA or RNA structure without breaking covalent bonds. Only affects hydrogen bonds. Renaturation. Annealing or spontaneous rewinding of the strands when the conditions return to physiological. 1. Her two-step reactions. In an experiment, you found that the treatment temperature is too high and denatures the DNA in your sample. What could you do to fix that? Increase GC content because it has 3 hydrogen bonds instead of 2, so it will be stronger. You have 5 samples containing the same amount of short, single-stranded human DNA in aqueous solution. To each of these samples you add one of these 5 short, single-stranded DNAs each of which is 100% complementary to the original. 1 human, 1 yeast, 1 mouse, 1 chimp and 1 chemically synthesized. How would their binding to the human DNA compare? They would be the same DNA as DNA. Hybridization utilizes complementarily hybridization techniques to detect specific sequences. Spontaneous mutations. Cytosine deamination 100 cell day, purine deamination 1 cell day, depurination 10,000 cell day. Induced promoted mutations. 1. UV ionizing radiation, pyrimidine, dimers, ring openings, covalent bond breaks. 2. Chemical agents. What chemical agents cause induced promoted mutations? 1. Deaminating agents, 2. Alkylating agents, 3. Oxidative damage by excited oxygen species. Deaminating agents is a chemical mutagen that can remove an amino group from a nitrogenous base, causes adenine to look like guanine. Nitrous acid bisulfate, used as food preservatives. Alkylating agents. Alter base structure, affecting H bonding. Oxidative damage by excited oxygen species. Damage caused by highly reactive oxygen molecules that steal electrons from other compounds, causing changes in structure and function. Hydrogen peroxide radicals generated during radiation or aerobic metabolism. DNA is the only macromolecule that has a repair system. Think of the importance. Methylation. A chemical modification of DNA that does not affect the nucleotide sequence of a gene but makes that gene less likely to be expressed, adds a methyl group. Sanger's method. Automated sequencing. All-in-one reaction, labeled DDNTPS, the use of fluorescently labeled deoxyribonucleotides and a fluorescence detector to sequence DNA. Each DDNTP is attached to a different fluorescent dye. Fragments move off gel in size order, pass through laser beam. The color each fragment fluoresces is recorded on printout. Phosphoramidite. Sequential addition of nucleotide units as nucleoside phosphoramidite derivatives to a nucleoside, covalently attached to the insoluble resin. Other functions of nucleotides. 1. Nucleotides carry chemical energy. 2. Nucleotides as cofactors, coenzyme A, NAD, PAD. 3. Nucleotides as messengers. 4. Functions of nucleotides. 1. Energy from metabolism. 2. Enzyme cofactors. 3. Signal transduction. 4. Building blocks of nucleic acids. 5 functions of nucleic acids. 1. Storage of genetic information. 2. Transmission of genetic information. 3. Processing of genetic information. 4. Protein synthesis. 5. Regulation of gene expression. What is a nucleotide? Pentose with a nitrogenous base and phosphate. What is a nucleoside? Pentose with a nitrogenous base. Nitrogenous bases are derivatives of pyrimidine and purine. Pyrimidine, 4-carbon 2-nitrogen ring. Purine, pyrimidine combined with a ring of 2 nitrogen and 1 carbon. Pentose in RNA. Beta-D-ribofuranose. Pentose in DNA. Beta-2-deoxyD-ribofuranose, O on 2 becomes H. Different puckered conformation of ribofuranose. C2 endo exo, C3 endo exo. Phosphate group in nucleotides. Negatively charged at neutral pH, attached to 5 position. Nucleic acids are built using 5 triphosphates. How many phosphate moiety do nucleic acids have per nucleotide? 1. Nucleobases. Derivatives of pyrimidine or purine, nitrogen-containing heteroaromatic molecules. Planar, almost planar. Nucleobases absorb UV light around 250 to 270 nat. Pyrimidine bases. Cytosine, thyma, uracil, all are weak H-bond donors and acceptors, neutral molecule at pH 7. Where are these found? Cytosine, thyma, uracil. Cytosine, DNA and RNA, thyma, DNA, uracil, RNA. Purine bases. Adenine and guanine are found in RNA and DNA, weak H-bond donors and acceptors, neutral molecule at pH 7. Prototropic tautomers are structural isomers that differ in the location of protons. What are 2 types of tautomerized nucleobases? 1. Cato-enol tautomerism, 2. Lactam. Both tautomers exist in solution, but blank forms are predominant at lactam tautomers at neutral pH, Cato tautomers at neutral pH. Absorption of UV light at 250 to 270 nanometers is due to pi bond electronic transition. Excited states of common nucleobases decay rapidly via radiationless transitions. In nucleotides, the pentose ring is attached to the nucleobase via N-glycosidic bonds. N-glycosidic bonds formed to the anomeric carbon of the sugar and beta configuration, position N1 for pyrimidines, position N9 for purines. N-glycosidic bonds are stable toward hydrolysis, especially pyrimidines. When are N-glycosidic bonds cleaved? When it is catalyzed by an acid. Free rotation to nucleotides. occurs around the N-glycosidic bond. Sequences of atoms chosen to define the conformation angle in purines is O4, C1, and 9C4. Sequences of atoms chosen to define the conformation angle in pyrimidines is O4, C1, and 1, C2. Angle near 0 degrees corresponds to which conformation? Sin, right above pentose. Angle near 180 degrees corresponds to which conformation? Anti, not above pentose. Anti-conformation is found in normal bDNA. Are nucleosides and nucleotides hydrophilic, hydrophobic, or amphipathic? Amphipathic. Solubilities: nucleobases, nucleosides, nucleotides. Nucleobase by itself is insoluble. Nucleosides are soluble because of O. Nucleotides are soluble because phosphates are ionized. When is modification done to DNA? After its synthesis. What is common in eukaryotes, also in bacteria? 5-methylcytosine. Epigenetic marker. Way to mark own DNA so that cells can degrade foreign DNA. Inosine. Found in the wobble position of the anticodon in tRNA, made by deaminating adenosine. Provides richer genetic code. Pseudouridine. Found widely in tRNA and rRNA, more common in eukaryotes but found also in eubacteria, made from uridine by enzymatic isomerization after RNA synthesis, may stabilize the structure of tRNA, may help folding of our RNA. Nucleic acids are bonded to each other by covalent bonds formed via phosphodiester linkages, negatively charged backbone. Is the DNA backbone stable, unstable? Stable. DNA backbone hydrolysis is accelerated by enzymes DNase. Is RNA backbone stable, unstable? Unstable. In water, RNA lasts only a few years. In cells, mRNA is degraded after a few hours. Linear polymers. No branching or cross-links. Directionality of nucleic acids. 5 is different from 3, and write and read the sequence from 5 to 3. Five and has a free Phosphate group not involved in ester bond. 3. And has free. Hydroxyl group not involved in bonding. Ester linkage in DNA goes from blank to blank. 3 to 5. What makes DNA stable? DNA lacks the 2O, so it lacks the nucleophile, which causes it to lack intramolecular attack, thus making it stable. Friedrich Miescher isolates nuclein from cell nuclei. Hydrolysis of nuclein of DNA. Phosphate, pentose, nucleobase. Chemical analysis of DNA. Phosphodiester linkages, pentose is ribofuranocide. Watson and Crick. Missing layer means alternating pattern, hydrogen bonding, A pairs with T, G pairs with C. Franklin and Wilkins. Cross means helix, diamonds mean that the phosphate sugar backbone is outside, calculated. Helical parameters for a single strand. What drives the formation of DNA? The hydrophobic effect. Watson-Crick model of bDNA. Ribofuranose backbone is on the outside, nucleobases are on the inside, stacked on each other. In DNA, two chains differ in sequence, and this helps with stability because O and phosphate group H is partially positive, thus neutralizing double negative on phosphate group. Run antiparallel. Nucleobases form pairs that yield the most stable pair, i.e., if you go against complementarity. Form the greatest number of H bonds between them. If you go against complementarity, you will have hydrogen bonding repulsion. Base pairing in regards to monomers and polynucleotides. Monomers, large number of base pairs is possible. Polynucleotides, only few possibilities exist. Complementary base pairs. A pairs with T, G pairs with C, purine pairs with pyrimidine. DNA double helix is stabilized by what two things? Base pairing, base stacking. A form DNA. 1. Occurs when DNA is dehydrated. 2. When DNA has a sharp bend. 3. When protein binds to DNA. B-form DNA. Most common form of DNA. Z-form DNA. Occurs when one protein binds to DNA. A form, sugar pucker conformation, major groove, minor groove, glycosyl bond conformation. C3, endocontract, major groove is narrow and deep, minor groove is wide and shallow, anti. B form, sugar pucker conformation, major groove, minor groove, glycosyl bond conformation. C2, endo expand, major groove wide and deep, minor groove narrow and deep, anti. Z form, sugar pucker conformation, major groove, minor groove, glycosyl bond conformation. C2, endo for pyrimidines, C3, endo for purines, major groove flat and wide, minor groove narrow and deep, anti for pyrimidine, syn for purine. Central cavity of DNA. Base pairing occurs around the central axis, hydrophobic layer surrounding central cavity. Central cavity of bDNA. Base pairing occurs across axis of symmetry, central cavity is completely hydrophobic. Central cavity of zDNA. Base pairing occurs around axis of symmetry, staggered, central cavity is hydrophobic. DNA denaturation. Covalent bonds remain intact, genetic code remains intact, hydrogen bonds are broken, base stacking is lost. Denaturation of DNA can be induced by high temperatures or change in pH. Can denaturation of DNA be reversible? Yes, annealing. Why does denatured DNA absorb more at 260? Because the nucleobases of the DNA aren't shielded anymore by the backbone, so it can absorb more UV light. DNA denaturation is monitored by UV spectrophotometry at 260 nano. The midpoint of melting depends on base composition. High CG increases Tm, DNA length. Longer DNA has higher Tm, pH and ionic strength. High salts increase Tm. Which base pairs melt at a lower temp? Melt at a lower temp than GC. Two near complementary DNA strands can hybridize, take DNA of two different organisms and get them to anneal. mRNA. Synthesized using DNA template, code carrier for the sequence of proteins. Instead of deoxyribose, mRNA has Ribose. mRNA contains what instead of thyma? Uracil. One mRNA can code for more than one protein. mRNA and tRNA transfer genetic information from DNA to proteins. Hairpin double helix. Complex structure of RNA are sometimes stabilized by non-complementary base pairing. Palindromic sequences can form hairpins and cruciforms. Palindromes. Words are phases that are the same when read backward or forward on two different strands. Molecular mechanisms of spontaneous mutagenesis. 1. Deamination. 2. Depurination. Cells have mechanisms to correct most of these. Deamination. Very slow reactions, large number of residues, loss of exocyclic amino group, 100 events day in a mammalian cell. Depurination. N-glycosidic bond is hydrolyzed, significant for purines, 10,000 purines lost day in a mammalian cell. Molecular mechanisms of oxidative and chemical mutagenesis. 1. Oxidative damage. 2. Chemical alkylation. Cells have mechanisms to correct most of these. Oxidative damage. Hydroxylation of guanine. Mitochondrial DNA is most susceptible. Excited oxygen species such as hydrogen peroxide, hydroxyl radicals, and superoxides arise during irritation or as a byproduct of aerobic metabolism, thus oxidative DNA damage. Chemical alkylation. Methylation of guanine. Molecular mechanisms of radiation-induced mutagenesis. UV light induces dimerization of pyrimidines. This may be the main mechanism for skin cancer, mainly between adjacent thymines on same strand of DNA, causes ring opening and strand breaking. Cells can repair some of these modifications, but others cause mutations. Accumulation of mutations is linked to aging and carcinogens. What end of the DNA strand is the primer and which is the template? Three. Strand is the template. 5. The strand is the primer. Next in DNA sequencing. 1. Template of unknown sequence. 2. DNA polymerase. 4. DNTPS. 4. DNTPs. 3. Dye-labeled segments of DNA copied from template with unknown sequence. 4. Dye-labeled segments applied to a capillary gel and subjected to electrophoresis. 5. Computer-generated result after bands migrate. Other functions of nucleotides. Energy source, regulatory molecules, coenzymes. What is a genome? The unique DNA content of an organism. An organism's DNA. What is genomics? The study of an organism's genome. What does genomics intend to do? Identify the encoding portion of genes. Identification of regulatory sequences for transcription. Identification of sequence for the initiation and termination of replication. Identification of intervening sequences for junk DNA. Genomics intends to identify the encoding portions of genes. This means what in particular? Genes for proteins, tRNA, rRNA, small nuclear RNA. Genomics intends to identify regulatory sequences for transcription, meaning promoter sites and operator sites. Promoter sequences. Identify the start point for transcription. Regulate the rate at which transcription occurs. Operator sites. Binding sites on DNA for proteins that are going to regulate transcription. A lot of what was thought to be junk in our genome actually encodes for microma. Microma play a role in the post-transcriptional regulation of gene expression through the process of interfering RNA. Proteins. Genes can be located on either strand of the DNA. Plasmid. Simple, circular DNA. In transcription, DNA serves as the template for the RNA being made. Can be any type of RNA, tRNA, rRNA, mRNA. During transcription, how is the template read? 3 to 5. During transcription, how is the RNA made? 5 to 3. Does genome size or the amount of DNA base pairs that an organism has have anything to do with the complexity of an organism? No, complexity is based on different number of cell types. Why did we sequence the human genome? We were looking to discover genetic mutations that would explain disease states. This led to gene therapy. Gene therapy. Find mutations in the genes that are causing disease and then replace that gene in an individual and cure that disease. We know of 50,000 minus 140,000 proteins, so we predicted that there were 50,000 to 140,000 genes. There is actually only 30,000 genes. Why? Alternative splicing. What is alternative splicing? Different forms of mature mRNA are generated from the same gene by splicing together of exons. We could produce related proteins with different functions from a single gene. Exons. RNA sequences in the primary transcript that are found in mRNA. Proteomics. The study of proteins. Genes and coding products required in large amounts may exist in multiple copies. rRNA, tRNA, small nuclear RNA. Most genes encoding for proteins don't exist in multiple copies, with the exception of histones. Histones are proteins that are required for DNA packaging. How are the genes in prokaryotes organized in genomes? Genes stand alone, can be contained in operons, organized in clusters, polycistronic mRNA, that contains the genetic code for more than one protein. What is an operon? Series of genes in tandem with DNA, and the transcription of all of these genes are controlled by a single promoter. How are the genes in eukaryotes organized in genomes? Genes can stand alone or organized in clusters, monocistronic mRNA that codes for only one protein. Histones are composed of 8 subunits, 2 each of 4 proteins. Each protein is an associated protein. These are all required to form a function oligomer that will help package DNA. Gene cluster. A tandem repeat of a single gene, separated by non-transcribed intervening regions, genes encoding proteins containing similar sequences or having related functionality in tandem and separated by non-transcribed intervening regions. Clustered genes may occur in no logical order and may be spread over multiple chromosomes. What is the difference between an operon and a gene cluster? Operons wouldn't have intervening non-transcribed DNAs, thus the genes would be next to each other. In tandem, a single promoter would regulate the transcription of all the genes. Arrows show the directionality of transcription, if they are all going in the same direction. They are probably on the same strand of DNA. Arrows show the directionality of transcription, if they are going in different directions. They are on different strands of DNA. Chromosomes can be adjacent in space, or they have the same regulatory mechanisms. What is a very rare way for the cause of a disease, and an example? Mutation of a single gene, X, cystic fibrosis. Most diseases result from the interaction of several gene products. There is mutation in several genes, it's these defects, and the protein products of these genes that result in disease. What are the genes called when there is only a single nucleotide that has been replaced or mutated in the gene? SNPs, or single nucleotide polymorphism. SNPs are responsible for susceptibility to disease, resistance to drug therapy, or adverse drug reactions. The knowledge of SNPs led to development of pharmacogenomics. Pharmacogenomics analyzes how genetic molecules affect an individual's response to drugs, and thus developing the most rational, effective strategy by using genomics and sequences of nucleotides. Repetitive sequences in prokaryotes. Non-transcribed regions usually are functional, such as promoters, ORAC, operators, etc. Selective pressure excludes junk DNA and for DNA to remain mutation-free. Repetitive sequences in eukaryotes. Apparently no selective pressure to restrict amount of non-transcribed DNA. Much of the non-transcribed DNA in humans consists of repetitive sequences of nucleotides. Repetitive sequences appear to be susceptible to mutation. What types of repetitive sequences do we have, and what are the two primary types? STRs, short tandem repeats, 1. Dinucleotide repeats, 2. Trinucleotide repeats. Dinucleotide repeats, genetically stable, have nothing to do with disease, don't have high mutation rate. Inheritable, characteristic of an individual's DNA, used for forensic scientists to identify suspects' DNA. Trinucleotide repeats, based on DNA fingerprinting, associated with disease, genetically unstable, associated with 14 human diseases. Age of onset and severity of disease correlates with the length of the repeats. Repeats expand with successive generations. Myotonic dystrophy, characterized by progressive muscle weakness and wasting. Autosomal dominant, it is a CTG. Repeat in the 5' untranslated region of the MPK gene expressing myotonin protein kinase, involves mRNA processing, has multi-protein, multi-organ system effects. Fragile X syndrome. Responsible for a lot of mental dysfunctions, mental retardation, CGG. Repeat in the 5' UTR of FMR1 gene, which expresses the FMR protein that assists in transporting some type of mRNA from the nucleus. FMR protein is heavily expressed in the brain and is required for formation and function of synapses. Huntington's disease. Autosomal dominant, characterized by cognitive decline, movement disorders, and emotional disturbances. CAG. Repeat is in the exon of the HD gene expressing Huntington protein. This exon is a portion of a gene that is both transcribed and translated. This mutation shows up in the actual protein itself. Effect of mutation promotes aggregation of the Huntington protein, leading to neuronal death. Why do we need to package DNA? To fit inside its membrane or cells, it needs to achieve a reduction in size. Supercoiling. Simple way to package DNA. Prokaryotes rely on this way to package DNA. Eukaryotes also rely on this way, but they also form specialized structures with the help of proteins in order to compact the DNA. What are the two types of coils? 1. Solenoidal coil. 2. Platonymic coil. Solenoidal coil. Much greater degree of compacting, adopted by an unwound DNA, much more efficient. Platonymic coil. A solenoidal coil folded on itself. We achieve supercoiling through the help of proteins. These proteins put solenoidal coils in our DNA and platonymic coiling in bacteria. Twist. Number of times single strands of DNA cross each other. Writhe. Number of times double helix crosses itself. If writhe number is negative, it is a negative supercoil, underwind and relax DNA. If writhe number is positive, it is a positive supercoil, add torsional strain. If we remove a couple of twists from a relaxed DNA, we will get a negative supercoil. If we add a couple twists to a relaxed DNA, we will get a positive supercoil. The simplest DNA an organism can have is circular DNA. The relaxed type of circular DNA has an ideal amount of twist and writhe in it. If we alter one, the other one also must be altered. If we alter one, but the other one is not altered, we are creating torsional stress on the DNA and the DNA will break. Linking number equals twist writhe. Are all structures with the same linking number topologically equivalent? No, linear DNA differs from closed, circular DNA. Are closed, circular DNA with the same linking number topologically equivalent? Yes. Are linear DNAs with the same linking number topologically equivalent? Yes. When torsional strain is put on DNA, the DNA will assume more stable forms. It can spontaneously go ahead and put in a negative supercoil, or it could underwind. Negative supercoiling does what to torsional stress? Relieves it. Positive supercoiling does what to torsional strain? Adds to it. In order to minimize torsional stress, DNA assumes a balance between negative and positive supercoiling in its structure. Consequences of underwinding DNA: only a few base pairs may cause strand separation, but because of the little amounts of base pairs, they will connect and cause a supercoil to get torsional strain again. Consequences of underwinding DNA: a lot of base pairs will cause strand separation, and due to the gap between the strand, a cruciform will form. Topoisomerase enzymes that are governed with making sure that DNA maintains the proper superhelical tension by controlling supercoiling. Topoisomerases are active during processes of replication and transcription. What are the two types of topoisomerases? Type 1 and Type 2. What is the Type 2 topoisomerase called for prokaryotes? DNA gyrase. What drugs inhibit eukaryotic topoisomerase? Chemotherapeutic agents. What drugs are DNA gyrase inhibitors, inhibiting type 2 topoisomerase in prokaryotes? Antibiotics. Type topoisomerase. Relax DNA supercoiling by cutting a single strand of DNA, passing the second single strand through the break and then resealing the break. Does not need ATP for energy. Utilizes energy in hydrolysis. A phosphodiester bond alters twist. Relax DNA supercoiling by breaking a single strand of DNA and then rotating the unbroken strand. The break permits rotation, does not need ATP for energy, utilizes energy in hydrolysis of phosphodiester bond, no passing strands through break, effect thrive. What does type of topoisomerase affect? Rive. Type 2 topoisomerase. Relax DNA supercoiling by breaking both strands of DNA, passing DNA through the break and then resealing the nicked DNA, requires ATP hydrolysis for the reaction. Rive is altered. Novobiocene. Topoisomerase 2 inhibitor, inhibits by preventing ATP bonding to the topoisomerase. Prevents interactions between the proteins that make up the histones, treats MRSA infections. Ciprofloxacin. Fluoroquinolone antibiotic, specific for DNA gyrase at therapeutic doses, going higher than therapeutic dose will start to hit type 2 topoisomerase, effective treatment for anthrax. Blocks topoisomerase from resealing the ligated DNA, left with DNA with breaks in it. Camptothecin. Chemotherapeutic agent, specific for type of topoisomerase, for bioavailability in humans, so derivatives are used. Doxorubicin, etopoxide, ellipticine. Specific type 2 inhibitors used in chemotherapy, to prevent topoisomerase from resealing the nicked DNA. Irinotecan. Type 1 topoisomerase inhibitor, derivative of camptothecin, used to treat colorectal cancer, preventing the topoisomerase from resealing the DNA. Topotecan. Type 1 topoisomerase inhibitor, derivative of camptothecin, used to treat ovarian cancer, preventing the topoisomerase from resealing the DNA. Chromosomes, prokaryotes and eukaryotes. A single molecule of DNA, an organism's genome, can consist of a single or multiple chromosomes. Prokaryotes have a single, circular chromosome. Eukaryotes have 46 chromosomes, 23 diploid. Chromatin. DNA that is bound to proteins that are necessary for it to be properly packaged. Prokaryotes do not have chromatin, only chromosomes. Eukaryotes. When we talk about chromosomes, we are talking about chromatin. Chromatid. Each individual chromosome. Centromeres. Each portion of the chromatids where they are joined together has a characteristic sequence of nucleotides associated with it. Telomeres. Region positioned at the ends of the DNA, contains special sequences of nucleotides that keep the ends of the chromosomes intact. Structure of chromosomes. If we were to denature the structure of a single chromosome, you would get beads on a string, beads. Proteins called histones with the DNA wrapped around it, string DNA. Core structure of chromosomes. Histone proteins with DNA wrapped around it, also known as nucleosome. What links nucleosomes? Linker DNA. What is a histone? Oligomeric protein, octamer contain 8 subunits, important for DNA packaging and gene expression. How does DNA bind to a histone? The DNA backbone has a negative charge, so the histones have to have positively charged amino acids on its surface. Only the surface interacts. Histone proteins can be reversibly, covalently modified by being methylated. If we take one of our chromatids and slightly denature it and cause it to slightly unwind, structure will open up and we can see the skeleton in the middle as well as the 30 nanometers fiber. N-glycosyl bond connects to 1 of pentose and either N1 or N9 of pyrimidine and purines. How is the N-glycosyl bond formed? Removal of elements of water. What is so special about pseudouridine in comparison to uridine? Uridine is connected to the pentose through N1, pseudouridine connects to pentose via C5. What makes the phosphodiester linkage? 5-phosphate of one nucleotide joined to 3-hydroxyl group of another nucleotide. The backbone of both DNA and RNA are hydrophilic, oligonucleotide, polymers containing 50 or fewer nucleotides. DNA double helix is held together by 2 forces. 1. Hydrogen bonding between complementary pairs. 2. Van der Waals and dipole-dipole interactions between stacked bases. Because of steric constraints, purine nucleotides are restricted to two conformations with respect to deoxyribose, anti and syn. Pyrimidines are restricted to what conformation and why? Anti because of the steric hindrance between the sugar and the carbonyl oxygen at C2 of the pyrimidine. Mirror repeat. Inverted repeat occurs within each individual strand of the DNA, do not have complementary sequences within the same strand, so won't form hairpins or cruciforms. What is an unusual base pairing in RNA? G combined with U and occasional A with T if there is residue. Predominant form of RNA double helix. A form right-handed double helix. What is a mutation? Alterations in DNA structure that produce permanent changes in the genetic information encoded therein. Reason why DNA contains thyma instead of uracil. The product of cytosine deamination is readily recognized as foreign in DNA and is removed by a repair system. If DNA normally contained uracil, recognition of uracils resulting from cytosine deamination would be more difficult, and unrepaired uracils would lead to permanent sequence changes. As they were paired with adenines during replication, this would also lead to a decrease in GC pairing and the genetic information attached to them. Examples of nitrous acid precursors and what do they take part in? Sodium nitrite, sodium nitrate, nitrosamine, accelerates deamination of bases. Examples of alkylating agents and what do they take part in? S-adenosylmethionine, dimethyl nitrosamine, dimethyl sulfate, nitrogen mustard, chemicals that damage DNA. UV light induces dimerization of pyrimidines when there are adjacent imines, creating cyclobutane-thyma dimer, which causes kinks in DNA structure and unmatched base pairing. In ATP, GTP, CTP, and TTP, the bond between the ribose and the alpha-phosphate is a blank bond. The bond between the other two phosphates are blank bonds. Ester bond, anhydride bonds. Hydrolysis of what bond on ATP, GTP, CTP, and TTP yields more energy? Anhydride bond. In RNA, base stacking interactions between what two nucleobases is the strongest? Two purines. Nucleosides of purine bases. Adenosine, guanosine. Nucleotides of purine bases. Adenylate, guanylate. Purines occurring in DNA nomenclature. Deoxyadenosine, deoxyadenylate, deoxyg. Nucleosides of pyrimidine bases. Cytidine, thymidine, uridine. Nucleotides of pyrimidine bases. Cytidylate, timidylate, arodylate. What is the general binding mechanism for proteins binding to DNA? Proteins are going to insert an alpha helix into the major groove of DNA, which is BDNA, protein. Binding typically induces conformational changes in both the protein and DNA that strengthen their interactions. What kind of DNA, protein stabilizing interactions are there? Ionic and hydrogen bonding between protein functional groups and ribose phosphate backbone of DNA. Van der Waals interactions between protein side chains and nucleotide bases, direct hydrogen bonding between protein functional groups and nucleotide bases, indirect hydrogen bonding between protein functional groups and nucleotide bases through a bridging water molecule. Bridging water molecule. The functional group on the protein and the base in the DNA are too far apart to hydrogen bond, so they hydrogen bond to a water molecule that is between them. One water molecule can hydrogen bond with 4 other groups. Indirect readout. Protein recognition of a specific sequence of nucleotides in DNA through the conformation of the ribose phosphate backbone and, or flexibility of this backbone. Different sequences of nucleotides will alter this conformation and give greater flexibility. May also be part of the process of molecular recognition for protein binding of DNA. Ionic bonding between protein and DNA. DNA interacting with the histone. Helix turn, helix. Most important motif in prokaryotes, alpha helices joined by beta turns. A series of alpha helices are set up so that alpha 3 gets inserted into the major groove of DNA, binding protein of alpha 3 is interacting with the nitrogenous bases. Zinc finger. Coordinate covalent bonds. One atom donates both electrons to form a covalent bond. Does the zinc finger require more energy than the HTH because they're making more bonds? No, they do not use energy to bind. If anything, there is energy released when they form hydrogen bonds with the DNA so that complex becomes more stable. What is the purpose of the zinc finger? Regulate gene expression, involved in gene replacement therapeutics. Leucine zipper. Repeating sequence of Lu mediates dimerization of DNA-binding proteins but does not bind to DNA itself. Lu acts as a hydrophobic zipper to join the proton subunits. Dimerization activates DNA-binding protein, only held together by weak, non-covalent London forces, making it easy to zip and unzip. Zipper straddles DNA in the major groove. Side chains from both helices extend into the groove and contact DNA bases through hydrogen bonds. Intercalating of molecules. What do they do? Molecules that can favorably interact with the nucleotide bases insert themselves between the stacked bases, distorts DNA conformation, hinders protein binding to DNA, prevents movement of a protein along DNA. Preventing movement of a protein along DNA can lead to problems because proteins move along DNA through the indirect readout mechanism. Intercalating molecules are blank and they can cause blank. Chemical pollutants, mismatched base pairing, used as chemotherapeutic agents. Chemotherapeutic agents intercalate into all cells, and that's where you have the bad side effects of chemotherapy. What are three typical intercalating molecules? Ethidium, proflavin, acridine orange. Human globin genes. Alpha subunit-like genes, beta subunit-like genes. They are located on different chromosomes. They are both in a cluster, will have their own promoters. We have different types of hemoglobin that is made up of different types of subunits as we go throughout life. Prenatal, alpha and gamma, postnatal, alpha and beta. Full expression of adult hemoglobin. Alpha 2 beta 2. Epigenetics. Modifications of histones in DNA that alter the structure of DNA may turn on or turn off transcription. Histone protein H1 binds to nucleosome and linker DNA and then binds to itself to form 30 nanometers fiber. What is 30 nanometers fiber? The solenoidal coil formed by the association of histone protein H1 to nucleosomes. What may also happen during indirect readout? It may become a part of molecular recognition for protein binding to DNA. The protein runs up and down the DNA. If it finds favorable interactions, it will bind. If not, it will repel and continue traveling down the DNA. If binding site it found, it will insert its alpha helix. Who donates what in the coordinate covalent bond? The CYs and the Hs will donate their 2 electrons to the empty orbitals of Cn. Ethidium. An intercalating molecule that has a phenyl group that sticks out of the DNA causing deformation and roadblock in major groove. Ethidium and acridine orange. Intercalating molecules, dyes that are used to visualize DNA and genes. All intercalating molecules can hydrogen bond, so why don't they? B, C of resonance. They are very weak H bonders, so instead they behave as hydrophobic molecules and thus insert themselves with the nucleotide bases. Actinomycin D. Chemotherapeutic agent, non-specific because it's an intercalating molecule, effective because no protein can bind to this, thus there will be no replication or transcribed mRNA.